And my name is Pratham Patel, and today me, Chetan, Geetika, and Ananya are going to present our poster presentation, which is on transcriptomic profiling of triple-negative breast cancer and normal breast tissues. Triple-negative breast cancer, or TNBC, is an especially aggressive form of breast cancer that lacks estrogen, progesterone, and HER2 receptors. Because of this, TNBC patients don't benefit from hormonal or HER2 targeted therapies, making treatment options limited and prognosis after worse compared to other breast cancer subtypes. In our project, we compared gene expression patterns, also known as transcriptomic profiles, between TNBC and normal breast tissues. The research focuses to identify key genes that are differently expressed and analyze the biological processes and pathways those genes are involved in. The expected outcome was to uncover molecular patterns that could lead to future therapeutic targets or research directions.
The objective of our study was to compare transcriptomic profiles of TNBC and normal breast tissues to identify differently expressed genes or DEGs and the biological pathways they are involved in. We hypothesized that TNBC samples would show a distinct gene expression pattern compared to normal samples, with significant changes in genes related to cell cycle, regulation, apoptosis, and immune function. To test this, we obtained RNA sequence data sets from the Cancer Genomic Atlas, the TCGA, and Gene Expression Omnibus, GEO. We assessed sequence quality with FastQC and trimmed adapters using Trimomatic. The reads were aligned to the GRCH38 human genome using the star aligner. We then used DE sequence for sequence 2 for differential expression analysis. A gene was considered significantly different if it had adjusted p-value less than 0.05 and a log base 2 fold change greater than 1 or less than -1. For function analysis, we used cluster profiler in R to perform gene ontology and KEGG pathway enrichment, helping us understanding the biological context behind the expression changes.
So our analysis revealed over 1,000 differentially expressed genes between TNBC and normal tissue samples. In Figure 1, the PCA plot clearly shows a separation between the two groups, confirming that they have distinct transcriptomic signatures. In Figure 2, the volcano plot highlights significantly upregulated and downregulated genes. Notably, genes like CCNB1, ORCA, and CDK1, all of which are involved in cell division, were upregulated. Meanwhile, BAX and CASP8, which are associated with apoptosis, were significantly downregulated. Figure 3, our heat map, displays the top 20 DEGs showing consistent expression differences between TNBC and normal samples. In Figure 4, the geo-enrichment analysis shows that upregulated genes are involved in biological processes like the mitotic cell cycle and chromosome segregation, while downregulated genes are linked to immune response and apoptotic signaling. Figure 5 represents KEGG pathway enrichment, revealing that TNBC samples are enriched in cancer-associated pathways like p53 signaling, PI3K ACT signaling, and cytokine-cytokine receptor interaction. And lastly, Figure 6 shows box plots for selected genes, visually confirming increased expression of mitotic genes in TNBC and decreased expression of pro-apoptotic genes compared to normal tissue.
So these findings support our hypothesis that TNBC has a distinct transcriptomic profile from the normal breast tissue. The upregulation of mitosis-related genes suggests increased cell proliferation, which is a hallmark of cancer. On the other hand, the downregulation of apoptosis-related immune response genes may allow TNBC cells to survive longer and avoid detection by the immune system. These results align with the previously published research on TNBC and offer potential molecular targets for further investigation. For instance, therapies that are targeting overexpressed genes like the ORCA or the CDK1 could suppress uncontrolled cell division in the TNBC patients. While our analysis was based on the public RNA sequence datasets and computational methods, it does highlight the power of functional genomics in cancer research. Future work could include experimental validation or expanding the study to compare TNBC with other breast cancer subtypes.